Hi, everybody. My name is Dr. Taylor Capizzello, and I'm here to talk to you guys today a little bit about brain waves and their generators. So today we're going to be going over an overview on this. And many of you guys, if you have already taken the QEEG course with InMindOut, you're going to see that a lot of the things we're talking about are also gone over in that for the brainwave aspect of it. And we're going to go a little deeper into that today. So the EEG is an electrochemical reaction, and it's predominantly oscillatory rhythms that are generated from different locations in the brain, and they serve different functions. These rhythms can be complex to describe and are going to become a point of confusion in our field, or they already are. They're generators. What the proven function of them are, visual examples of how we see them in a 19-channel set, and how we see them and how we often link it to in our field is going to be gone over today to try and pull things together and make a little bit more sense. We're not going to be able to go over each one in each example, but we want to provide a reference sheet to see how members of our community may use these rhythms and linking symptoms with the EEG. In our field, we look at the phenotype and train that, not the diagnosis. Many diagnoses will have crossovers in the EEG pattern, so although it is helpful to connect the dots, ultimately the important part is looking at what we expect to see in the EEG and what we actually see and help that person reach their goals to attain that. You might have seen this before. If not, it is a really good slide, and it goes over all the different neurotransmitters and areas of the brain that are involved. So when we look deeper at this, it really tells us everything. And while you're taking a look at this, I'm going to read a quote from Niedermeyer. So various patterns of the oscillatory activity develop in cellular brain networks and are further reflected at the EEG level. The EEG is essentially an electric phenomenon. Therefore, we're going to focus on the ionic currents active during various oscillating patterns, but we're also going to identify the minimal anatomical structures necessary and sufficient for the genesis of the aforementioned activities. I want to first start by saying that this whole talk came about because of a group that we were reading, Niedermeyer, and we each were looking at components of the chapters and trying to learn from it. And I chose this chapter looking at brainwaves and their generators because I really wanted to better understand it. So I come at this from somebody who has taken a deep dive into this. I would not claim being an expert on this, and it's constantly an emerging field, and there's lots of information. So I wanted to start creating basically a folder of all the information that we have on each one. So when we hear conflicting information, we can come and see that. So that's what this is starting to be a process of, and it can be built on from here. First, we're going to dive into delta. So the origin is the neocortex, thalamus, basal ganglia, and it's responsible for slow wave sleep, deep non-REM sleep, signal detection and decision-making, memory consolidation, concentration, motivation, focused attention, and facilitation of intralaminar interactions in the cortex to control synaptic rescaling. One of the things I really like about this and the format that we're going to go over is we're going to talk about what science is showing for these waves, because a lot of times in our field, we don't think about delta as being concentration and motivation. But when we actually start looking at it in healthy controls during the tasks that delta is emerging, it's happening in these times. We're just not always going to see it. So once again, tying in what the science is and what we're seeing together. The generators are an interplay between low threshold calcium transient current and hyperpolarization activated cation current. The NMDAR-driven depolarization of an intrinsically bursting neurons, and the neuron glial interactions to regulate intracellular potassium through calcium waves. One of the important things to note about delta is that it's a cortical rhythm generated by sheet dipoles. Subcortical structures may turn off or turn down the input and allow delta to emerge, but it is cortical. Just like we're going to talk about later, beta and gamma. So delta is actually cortical. It appears to originate in layer 5 of the cortex and correlate with reduced pyramidal cell activity. There are two general patterns of the EEG seen: cortically generated delta, which is a disconnection of the cortex from the thalamus and results in increased delta. That's what we'll oftentimes see in our practice. And then we have the thalamic-cortically generated, which is if a person is sleeping, it's going to be thalamic-cortically generated. So we're usually not going to see that in our practice. Delta S pathology is when it is continuous polymorphic delta activity. Both metabolic and structural pathology can create it. Or if it's intermittent, often paroxysmal monomorphic delta activity, which tends to be prefrontal predominance, also called FIRDA, for prefrontal intermittent rhythmic delta activity, can also be posterior or focal, and then it's called TERDA or ORDA. So how do we see this in our field? In our field, we're going to see it normally in infants and very young children, which we might not be working with, but if we were, we're going to see a lot more delta activity. We're going to see it in slow wave sleep, which once again, we usually don't see, but every now and then your client might actually completely fall asleep, or if you're working in a sleep lab. Usually we'll see it in a TBI due to damage on white matter. It can be either focal or coup contra coup, and that's when a person hits their head in one spot, but then the brain smushes and hits the head in the other. So we're going to see one spot and the other of that delta slowing. If it is focal, we're going to want to consult with a neurologist to rule out other focal disturbances. We always want to stay within our scope of practice and make sure that we have another set of eyes on it, because it might be something bigger, and neurofeedback does not fix everything. It's also, we'll see it diffuse, seen in certain types of dementia or metabolic, endocrine and adrenal issues. It can also be linked to demyelination, hypoxia, and poison. And finally, we'll see it as a K complex when a person is dropping into stage 2 sleep. So at that point, it's going to look like a delta wave, and it technically is, but we're going to talk a little bit more about that later. I include it because Niedermeyer includes it in as the delta. I always like to add a little extra because when I'm going through this, I feel like there's more things that we need to know about that. And so this is also the area that I really want to grow within our field and keep adding into it how other people see it, and either cite that or cite the research that's currently being done on it, or if it's a hypothesis. So delta does play a role in the release of certain hormones, which could be a reason why when we see low voltage, slow EEG, it can indicate an issue. And a lack of delta waves, especially during sleep, can lead to issues as well. So everything we want balance. We don't want too much of, we don't want too little of, and that's going to be the theme for every single wave. I feel a lot of times we start to learn these and we don't get to see enough examples. So this talk is going to have about half, maybe more of the slides are going to be examples so you can see it. So here we can see the delta. It is a focal delta, and there is some slow background as well. And here is the example of FIRDA, like I was talking about earlier. One of the things to note with FIRDA, I talked about FIRDA, TERDA, ORDA, is that two of the delta rhythms correlate with age, FIRDA being more common in adults and ORDA being more common in children, so the occipital. And FIRDA is regarded as a projection rhythm. It may result from pressure on a deep midline structure, such as that associated with a mass in the third ventricle or posterior fossa. However, it is clear that FIRDA can also occur in a large variety of other neurological disorders and disturbances, including structural brain lesions, diffuse brain injury, metabolic dysfunction, and postnictally. Here we can see we are not looking at our normal average montage or linked ears, but actually, it is set up so we can source it. And you're always going to want to change your montage to, especially if you see slowing, you see something, you want to play with a couple different montages. And here we're able to see that this is frontal interrhythmic delta activity. So at this point, this client was referred to a neurologist. They did have a tumor. Here's an example of TERDA, which is the only one linked to epilepsy. TERDA is a localizing value indicating a dysfunction of the underlying temporal lobe. It is often interictal finding in persons with focal temporal lobe epilepsy. So you might see this and not see any epileptic cord discharges, but it's a sign that something deeper might be going on there. Once again, refer to a neurologist. They can do a longer EEG recording and see what's going on. Here's an example of delta at the vertex with global delta. So it's pretty clear at CZ when we look in the center that we can see that slowing, but we also see slowing throughout the record. And we're going to look at it in spectral next. So next we can see it with that vertex. and also additional slowing throughout the record. This is frontal rhythmic slow burst, and it happens during drowsy, and it can be a normal variant for teens and younger, and on rare occasions adults. This particular example was from an adult. It's called hypnagogic hyperosynchrony, and it can present in sleep disorders such as night terrors. It's non-specific variant, and it often corresponds with individuals that experience parasomnias, which are behavioral sleep disorders. So something like sleep terrors, sleepwalking, sleep talking, behavioral REM, where you actually act out your dream, sleep paralysis, and more. So when you see this in a client, once again, we're not trying to fix this, but it's going to flag they probably have night terrors or sleep paralysis or something like that. So don't be alarmed sometimes, and usually you can talk to your client and they're going to be able to confirm this. And finally, like I said, with the K complex. K complex is responsible for sleep deepening, information processing, and homeostasis of synapse and memory consolidation. A K complex is a response to a knock and is comprised of two things. It is an event-related potential, ERP, likely the auditory P100, seen at the vertex, because that's where we're going to it's going to come up from here, and sleep spindles. And this keeps the person from alerting as they're dropping into stage 2 sleep. It's in the delta band because most ERPs are going to trigger a delta band filter. And there is a difference between spontaneous EEGs and event-related potentials, but this is included in the summary in the raw waveform. It is a singular event. It's not a delta wave rhythm, but within the frequency. And I'm going to show you why in a little bit. So first, how we see it in our field is when a person drops into stage 2 sleep. This is one of the things that you will see in your practice. And that's one of the other reasons why I wanted to include it. When a person has their eyes closed and sometimes open, if we're seeing this, if we see it anytime before 300 seconds or 5 minutes with eyes closed, we want to refer them to a sleep lab because a person shouldn't be dropping into stage 2 sleep under 5 minutes sitting in front of you with their eyes closed. Or at any time with eyes open, we also want to refer to a sleep lab. Something else could be going on. We want to have it ruled out like sleep apnea. Once again, not something that we're going to work with neurofeedback, but it's going to really be causing problems for them. And what's that look like? Here, this person had 3 right in a row, so they were a really good example and really easy for us to see. Now, if they go to a sleep lab, it's going to look a little different, and I wanted to also show what it looks like for a sleep lab. So we can see here these much sharper waves that occur for what they're seeing, because they're looking at it much faster. So everything's very smushed together. Oh, I thought that I was going to be able to go over showing what it looks like, but it will actually look at in the spectral, we can see it, once again, looking like delta. And we want to make sure that if we see that, we can go back and look, oh, is this a sleep vertex, K complex, or is it actually delta at the midline? So once again, when we see in the spectral, we want to go back and know what we're seeing and why. On to theta. The origin is the hippocampus, dendrite gyrus, cortex, amygdala, and it's responsible for REM sleep, selective attention, arousal, orienting, voluntary control of movement, modulation of synaptic strength, and coordination of phase coding, of active neuronal ensembles, episodic memory, word integration, and environmental encoding. And I know you guys can all read this, but I think it's so important to really take in all these things that these waves actually do, when in our field, we really don't think of them this way. Prior to really going into this, I didn't really think about theta as episodic memory, but it is, and we're really going to go into that in a little bit. The generators are the interplay between inhibitory and excitatory hippocampal neurons that is modulated by cholinergic and GABAergic input from the medial septum. We're going to go over that on the next slide, or next 2 slides. Interplay between slow inward potassium currents and persistent sodium current. Acetylcholine-mediated calcium release from the astrocytic internal stores, and neuromodulation of the prefrontal cortex by dopamine. It actually is pretty complex, and we'll see that soon. So the limbic system generates theta by having the nucleus accumbens provide acetylcholine to the temporal lobe via the diagonal band of Broca. And thus we can see midline theta activity frontally. but also in the temporal area. So I want to repeat that because that's a mouthful and it's pretty cool when you think about this. And this came from Yuri Kropotov. So the limbic system generates theta by having the nucleus accumbens provide acetylcholine to the temporal lobes via the diagonal band of Broca. And that makes it so we see midline theta. That is where it's coming from. But we'll also could see in the temporal lobes because of that. We have normal theta waves, rhythmic midline theta from the limbic system. And Niedermeyer classifies these two types of hippocampal theta, one atropine sensitive and the other atropine resistant. For type 1 hippocampal theta, the most widely accepted hypothesis proposes that the frequency is determined by the feedback loop involving the medial septal area in hippocampus. And I say this a hypothesis because we still don't know. We still don't understand all of this, but this is what the leading research is, although there is some other research out there as well. For type 2 hippocampal theta, the supramammillary nucleus of the hypothalamus appears to exert control. So rhythmic theta is limbic theta. Then we have pathological theta waves. And pathological theta waves are either going to be slowing down of the alpha band intermittently and with frontotemporal predominance, which is attributed to disturbances in deep midline structures, or localized theta activity may merely represent the mildest form of polymorphic delta activity. So once again, we're hearing theta, and actually pathological theta tends to be slowed alpha or polymorphic delta activity. And we do have theta in there as well. But this is because of a relationship between the EEG frequency and the rate of cerebral blood flow and oxygen uptake in cortical gray matter. The slower the frequency, the greater the reduction in cerebral blood flow. We're going to go over this later as well when we talk about blood perfusion. And this is linked to moderate hypoxia, cerebral vascular disease, dementias, and mild degrees of metabolic encephalopathies, such as renal or hepatic failure. So, thinking about theta, is it theta or is it slowed alpha? Is it polymorphic delta activity? For healthy theta, theta is generated in the septal hippocampal network with excitatory and inhibitory interconnections. We're really going to go over a lot of inhibitory, so be prepared. In a resting state, the hippocampal pyramidal cells are inhibited by a local inhibitory neurons, and the gate to the loop is closed. An important event activates the septal inhibitory neurons, which inhibit the inhibitory hippocampal neurons and thus remove inhibition and open the gate. So lots of inhibition for this burst. The cycle is repeated by the reciprocal excitatory inhibition hippocampal septal connections. And this burst is what happens, this under 1 second burst of theta activity that we see. And this is the one that is linked to memory and everything else that we were talking about, that it's all these really good things that we often overlook for theta. Theta should come in a burst under 1 second, and during our recording of eyes open and eyes closed, we do not expect usually to see too much of this because we're not giving tasks, but if you are doing a task, then we will see more of this, or if the person just randomly has this a couple times. So we can see this when we look here, that that burst of theta activity into at the midline. Remember the diagonal band of Broca that's happening, and we see that burst that occurs there. But in our field, how do we usually see theta? And it's usually when a person is daydreaming or tuning out, or they're tired and drowsing. And remember, that's probably alpha, slowed alpha happening because they're getting tired. We see it a lot as prefrontal in people with ADHD, depression, anxiety, OCD, and we see it as persistent, rhythmic, slow wave. Theta can be present in TBIs, and it's often seen as focal or coup contra coup, just like we did at delta. For this, it's when there is gray matter damage. So white matter damage for delta, gray matter damage for theta. And we're going to see it in tonic brief bursts of theta at the frontal midline increased with task load, and it is a normal memory function. Here's some pathology of theta examples, and we can see pretty clearly a lot of slowing going on. So we can look here, and we can also judge that some of that might be that polymorphic delta activity. Here's the burst of theta mixed with that slowing of theta background. So we have a burst, normal, but then we also have that slowing, and we can see that it's very different. and we look at the frontal pathology, we can see our alpha, and then we see the theta in the red bands happening. We see a possibly slowed alpha and an additional theta band right there. Here's another example, same thing, just different person, that theta that we see frontally. And here's a mix of delta and theta as well. So we can have a mix of this a lot of times in head injuries at sea, or any sorts of ischemia. And this is what it looks like. So we see a lot of slow activity happening. This is alpha slowed into the theta band. If you're familiar with looking at the raw EEG, we obviously see this as alpha, but a computer might not, because when we switch over here, this is actually slowed into the theta band, and it can get even slower than this as well. This is just a nice one that you can see how much it's slowed. And this particular person has migraines because we have that slowed alpha in the temporal regions. A little extra, frontal theta is a good responder to stimulants and only has about a 15% success rate with SSRIs. So if your client has a lot of frontal theta, SSRIs might not be the choice for them. So far, the only research out there is for ketamine. That is from Jay Belkelman. And stimulants usually are good because we're waking up the system with that. So they can be good responders for it. Lambda is in the theta band. So just like we talked about the K complex, we're going to talk about lambda, which is also called lambdoid and post. It is seen occidentally at sites O1 and O2 as triangle waves with a sharp positive phase. And it's seen when someone is scanning, they're visually tracking their eyes. You will not see their eyes move. Well, at least our trained eyes won't see them move. But when we look at spectra, it can look like theta coming from the back of the head. And in eyes closed, imagery elicits lambdoid activity. And in stage 2 sleep, it's called post for positive occipital sharp transients of sleep. Mostly, we're going to see this in 6 months to 10 years old. So a lot of your kids are going to have more lambda. It is called lambda because of the sawtooth shape that resembles the Greek letter lambda. We can see in this example, these sharp, more V shapes that are happening. And I just had a client who I was going over whose whole background activity was all lambda. And if I had more time, I probably should have put their example up. And it's commonly seen in, like I said, people with PTSD because they're just constantly tracking. So now on to alpha. So alpha is the background rhythm, and we're going to talk about it. Its origin is the neocortex and the thalamus. Thalamus we're really going to talk about. It's responsible for drowsiness and relaxation, sensory function, movement, and visual perceptual framing, task engagement, speed of working memory, and cognitive performance. And the generators are cholerogenic modulation of the prefrontal cortex, activation of metatropic glutamate receptor in the gap junction, high threshold bursting neurons in the lateral geniculate nucleus, and activation of the noradrenergic neurons in the locus coeruleus, mediated by corticotropin releasing hormones. So just like I was saying, it's the background rhythm, and it increases with age, peaking around 9 to 10 years old, and it will be around 10 hertz. And around age 65, it starts to decline, 0.5 hertz a decade. So we're going to see it start slowing down. And it's defined by the morphology being typically rhythmic and sinusoidal, just like we saw in that example of slowed alpha being in the theta band. We have that really sinusoidal rhythm. Alpha and spindles are often confused. There's a lot of this in Niedermeyer about it, so I do want to spend just a little bit on it, and I hope it doesn't feel too confusing. So spindles are going to be talked about more in beta 1 as it is in beta 1 band. Alpha waves are mainly distributed along cortical pathways, whereas spindle waves are generated in the thalamus and exclusively distributed to neocortex among thalamocortical axons. An important role to these thalamic nuclei is the cholerogenic input that arises from the brainstem and basal forebrain nuclear groups. It has been shown that cholerogenic inputs are excitatory to the thalamocortical cells, causing a direct depolarization associated with a decrease of potassium current. At the same time, there's a decrease of the inhibitory input arising from the thalamic reticular neurons. And since the latter are hyperpolarized by cholerogenic activation of a potassium conductance. And in this way, cholerogenic inputs favor the blockage of rhythmic spindles, a phenomenon that is generated in the thalamus and shares the same frequency range with alpha oscillations. So once again, computers don't know the difference of this, but they are the alpha and spindles, even both coming from the thalamus, are going to be different. We're going to see them differently. They have different pathways. So our eyes might not know that, our computer might not know that, but just kind of having that bit of knowledge to be aware of. Thalamus is really the generator for so much stuff, right? So alpha, we'll talk about, because that's our topic right now, is generated from an electrochemical change in one area of the thalamus, and it moves from that area to other nuclear bodies in the thalamus like a wave. And this phase of the alpha sweeps through the thalamus. And then this projects to other areas of the brain, and it should present back to front. And we see this as it moves across the cortex in the EEG. And the phase of the alpha is going to be related to the phase of the generators. And in a little bit, we'll dive into this part a little bit more. When we're looking at alpha, the electronegative alpha is open, seen as up in the EEG, and that's when information is coming in. And the electropositive alpha, the gate closes momentarily, and that's seen as down in the EEG. So these information packets that are happening. So when we talk about our processing speed of alpha, we talk about these information packets that are going through the brain to give information. That's where this is coming from. That said, when we have higher amounts of alpha present, that area is in a resting state, and that's going to be talked about in the next slide. We have lots of different networks that are important, but in our field, we really focus on the default mode network and the salience network, which then allows us to get to the executive network. When alpha is present at the back of the head, it's taking a snapshot of what is perceived, and then it turns off. And that packet of information sweeps up to the front of the head to be then evaluated and processed elsewhere. And the front and the back of the head are going to be out of phase. And this allows a person to take in information and process it at the same time. Because of that, we know alpha is not asleep, but a resting state that is active and ready. And this information is part of a phenomenon of traveling waves and helps the brain process information appropriately. So this is why we can do so much so quickly, because as we get information, it's processing it in the front and we're getting more information, and it's just keep coming in that way. So the default mode network is parietal by itself, PZ, then PZ to the left, then PZ to the right, then PZ to FZ to the front. And it's unidirectional. So it's going forward in one direction, and then PZ by itself. So when we look at an fMRI, PZ looks so strong, but that's just because it's been hit so many times by this information. And if it sends information to the front and the salience network turns on, then that salience network is going to turn the information off. And we turn off that default mode network so the salience network can be on. So here it's going from the front to the back, turning off the default mode network. And this is a resting state that is ready. So when there's something salient, it can then activate. And so this is why PZ is part of the Rich Club Hub, because it is just at the center of everything, making it that a person, if everything's acting correctly, is checking in with all parts and then turning off and allowing the salience network and then going into the executive network. So when we work with clients, we really want to have this nice connection going between all of them. So if the salience network is turned on permanently, then we're going to end up with a low voltage fast EEG, super hypervigilant. Since everything is salient, you're always alert and you don't come into that resting state, that default mode network. And the salience network inhibits the default mode network, and then the default mode network can inhibit the salience network. So that's when the salience network is kept on, the background activity drops out. And if the default mode network is stuck on, then the salience network can't operate, and the person would be present as inattentive and unable to focus. So once again, that Goldilocks, having that balance of everything. As a side note, if you have a lot of alpha in the salience network, it's probably not on, because the salience network is going to be activated by beta or gamma. Another side note, in pain, the salience network is called distress network. And it's same thing, distress, salience network, because there's nothing more salient than distress. And we can see in the location of the salience network that the anterior cingulate cortex is associated with it. So if you have distress from your tinnitus and your intercingulate cortex locks onto your tinnitus and your temporals, you're going to end up amplifying that sound with your attention. Because when you pay attention to a stimulus, you augment the perception of it or you potentiate it, and you sculpt what you perceive for your attention. So tinnitus becomes greater and greater because of this. Alpha is also an attention mechanism. And it is because it's a generally inhibitory mechanism of the brain. So as with these studies, it shows that alpha increases to use to create increased focus in other areas needed for attention. So my hobby is I'm a beekeeper. So I end up getting lots of speakers for meetings that are all beekeepers. And I end up getting to learn so much. And bees and brains have so much in common in how we study it. So they had actually done a whole bunch of studies looking at bees and their alpha waves. And it really started showing us how much we use alpha for our attention. So an easy example for us is thinking about if we're driving down the road and there's a dog on one side of the road, not leashed, and there's a car just coming regularly on the other side of the road, we want to pay attention to where that dog is. And so our alpha on the opposite side is going to increase. So our attention can focus on that dog more. We're kind of put blinders on everything so we can bring it. So alpha can be really good for that. Once again, we want everything to be flexible. We want to have increases and decreases. But in our field, we don't want to see anything stuck on. What do we expect to see is when a person closes their eyes, we expect to see alpha parietally at PZ 01 and 02, and a little bit as it goes, but getting lower as it goes to the front. And it's commonly seen higher at site 02 than 01. And when a person opens their eyes, we expect alpha to decrease by 50%. And when we see it dominant in other areas, it can signify a dysregulation. That area is not functioning as ideally. We're not getting as much blood flow to it, and we're going to talk about that in several slides. In our field, we're going to see it as a posterior dominant rhythm, PDR. We're going to see it prefrontal. It can be linked to depression or anxiety, with the latter more likely of hypercoherence of alpha is noted. Balance of symmetry of alpha in different locations or slowed alpha can mean several things, including dementia, ADHD, trauma, ischemia, and on. It tends to be higher over the right, the non-dominant hemisphere, if there isn't much alpha present due to alpha being very fast or no alpha at all. It is a low voltage fast EEG. So if we don't have very much alpha there, it's, and it's super fast, we're gonna have a low voltage EEG. This presents as over aroused due to being inundated with information. And it's seen in those with a genetic marker on gene 4, which controls GABA A receptors. If you make more receptors, you can't saturate them and you're over aroused. And it's common in people with alcoholism and PTSD. So that lack of alpha can be linked to it. Once again, we always want that Goldilocks, a nice balance. We do want some alpha. We want it to be able to turn on and off. Whatever area we see excess alpha power over the brain, we can guess that they're going to be struggling in the areas of the brain that it's over. And it's also in somatic and declarative memory. Here we see frontal alpha and hypercoherent and varietal alpha. So this is eyes closed, but we can see it's pretty strong. And if we look at it this way, we can once again see that's pretty high up. So that person has a lot of hypercoherence. Here's alpha slowed into the theta band. This is the same example we showed before. So we can see that it's still alpha, but it's been slowed down. And here's low voltage fast eyes closed EEG. So this person has no alpha, even with their eyes closed, and it's very fast. And here's what that looks like in spectral. So really no alpha present. This person does identify as an alcoholic, which makes sense given the EEG that we see. Well, extra things, THC can be shown prefrontally as hypercoherent alpha or excess alpha. So that other person that we saw earlier that had a whole bunch of prefrontal alpha, they either are using a whole bunch of THC or we wouldn't want them to because it's going to exacerbate that even more. People with frontal alpha have been shown to have a 85% success rate with the use of SSRIs, but that's only if their alpha is a little under 10 hertz. If the alpha is faster than average, especially above 12, it can result in anxiety as SSRIs speed up a person's alpha. Mu, which is an alpha band, which is totally different. It comes from different areas. It comes from the somasensory parietal cortex. It's involved in frontal lobe disengagement of the mirror neuron system and somatosensory information. Mu is in the alpha band, but it is an arc form or cone-shaped rhythm, and it also decreases as a person becomes drowsy. So we're gonna, I'll talk next about how we see it, and it's pretty easy to see here. We are going to usually see it. It's about 0.5 to 1 hertz faster than the parietal alpha, but I honestly see it slower plenty of times. So it's just going to be different than the parietal alpha. It's seen at C3 and C4. Sometimes we're going to see a little bit behind parietally. That will occasionally happen. It's a disengagement of the frontal lobe's mirror neuron system, seen with those that tune out or are trained to tune out, such as athletes, seen in 50% of the autistic population, 50% of attention population, 15% of normal population, which includes a lot of CEOs and athletes and people that are trained to tune out. And we're going to see it when oxytocin is present, mu is removed, and eye contact produces oxytocin for most people. Here we can see it. It's pretty obvious. Our eyes are drawn to it. Sometimes I love seeing mu in the record because it just looks so cute to me, like little M's across. And here we're going to see it at C4 as that spike. And it's, we can see that it comes directly from that. And it's different from the person's alpha that we see at 10.74 hertz. And for this person, it's faster. On to beta. How are we on time? Okay, I got so much stuff. Like I said, there's a lot of things in here. But so beta comes from the neocortex, the olfactory bulb, striatum, and the thalamus, hippocampus. It's involved in sensory motor control, motor preparation, sensory processing, amplification of olfactory and visual stimuli, obviously not something we're usually going to see, top-down attention, and working memory allocation, of which we do. The generators are the activation of gap junction coupled layers 5 neurons mediated by the M-type potassium current, acetylcholine modulation of synaptic interactions between layer 5 pyramidal neurons, and low threshold spiking interneurons. Beta waves are often considered indicative of inhibitory cortical transmission mediated by GABA, the principal inhibitory neurotransmitter. The blockage of GABA receptors enhances beta. So the blockage of GABA receptors enhances beta, while it will abolish gamma rhythms. They are not the same. Beta and gamma oscillations rely on different mechanisms, and the gamma rhythmic activity of the superficial layers and beta rhythmic activity of the deep layers can be cut and still maintained separately. So they are very different. Go a little bit more into that. In the literature, beta and gamma have several crossovers, including playing a large role in the blocking of slow waves. The cellular mechanism of this suppressing mechanism includes the blockage of spindles occurring at the very site of their genesis, the reticular nucleus, through the action of acetylcholine, serotonin, and norepinephrine. And remember that slow alpha and theta waves can be altered by these neurotransmitters. The intrinsically generated thalamic delta oscillation is blocked through the depolarization action of both acetylcholine and monoamines on thalamic cortical cells. And finally, slow cortical delta activities are prevented by cholinergic actions of the nucleus basalis neurons. And remember that delta from the thalamus is related to sleep. The synchronization of beta-gamma activities strictly confined to synaptic neurons, achieving a more specific aggregation of the neuronal populations involved. So lots of similarities, lots of differences, and totally different generators. Remember, they can have everything cut and still maintained. We have different types of beta. We have frontal beta, which is fairly common, central beta, and Rolandic beta, which is obviously central. It can be blocked by motor movement. It's generated in the basal ganglia and thalamocortical loop. It's partially, but not generally, the basis of Rolandic mu rhythm. Posterior beta and posterior rhythms, often a fast alpha equivalent, but in the beta bands, it may be considered fast alpha, and it may be linked to hyperthyroidism. Diffuse beta, seen throughout the recording. Vertex beta, fast central midline rhythm around 25 hertz. This is the wakefulness drive, commonly seen in insomnia. We have Rolandic cortical area, which is 15 to 30 hertz. We really don't ever talk about this, so I did want to include this one. Like I said, anything in Niedermeyer that it wasn't something we talked about, I really wanted to make sure I include it as well. Displays a close temporal relationship with peripheral EMG activity during isometric contractions. And this implies that this beta rhythmic activity is associated with rhythmic firing of the neurons of the motor cortex that generate the command to drive spinal motor neurons. On the other hand, there are other conditions where beta rhythms are not associated with a state of active neural firing. So when we see that EMG activity, there might also be some underlying truth to some of the beta. In our field, we really talk about beta 1, which is 12 to 16 hertz. It's more about a calm, stable focus. Beta 2, 16 to 20 hertz, the intense problem solving or action. Beta 3, 20 to 30 hertz, high beta, busy brain, rumination, or an agitated cortex. And the fourth type is the shear rhythm, 30 to 42, can be important for learning, also called a buying rhythm and seen in peak performance. In our field, we're going to see an excess of beta in an agitated cortex. So we're going to see it in rumination and anxiety in beta 3, linked to OCD, ODD, amotivational when seen at the anterior cingulate cortex. 20% of ADHD people have frontal beta spindles. In CEOs and athletes, we can see this at CZ and or FC as a driver. Frontal beta spindles are seen in bipolar. We see it in insomnia as spindles at CZ and or FC. And there's a hypothesis that if diffuse beta activity, there may be inflammation somewhere in the body. Symmetrical beta is usually during light drowsing, and asymmetry greater than 35% is abnormal and could have underlying issues. So that's for beta. Here we can see beta at the vertex at CZ. It's pretty easy to spot there. So this is someone with insomnia. We can see that as a little blue bump, and obviously it's not going to have a lot going because it's beta, and beta is always going to be smaller. So you don't want to see a high peak like we would in alpha. This is diffuse beta, so we can see beta throughout the entire record. So this could be a candidate for possibly having, inflammation somewhere else in their body. We can see that with the beta spindle at CZ and diffuse beta throughout. Here's F2 beta spindles, giant sinusoidal beta. This is a client that with ADHD, and we can see very clear frontal beta spindles, not muscle related. Here's beta gamma. So we're getting into, or some people call it gamma. So very fast activity going there. It can also be seen from medication in the lower beta band, such as benzodiazepine. So if we see diffuse beta spindles, we're going to also want to look at what medication they're on. Beta amplitude and quantity is enhanced by bone deficits, including skull breaches. So if that person actually has a breach at the skull, we're going to see, could be seen changes in beta activity. On to SMR, we have that's the spindles, also called sigma, also called sensory motor rhythm, also called sleep spindles. So the origin is the thalamus and the cerebral cortex. It's responsible for stabilizing rhythm, optimal blood flow, and staying deeper in sleep by blockages of incoming stimuli on their way into the cortex. The generators are the dorsal thalamus, made of several nuclei, each of them containing both relay, thalamocortical, and local circuit interneurons. The reticular thalamic nucleus generates spindle rhythms. The reticular nucleus is a purple sheet of GABAergic neurons covering the rostral, lateral, and ventral surfaces of the thalamus, and the thalamocortical neurons display rhythmic, long-lasting hyperpolarizations. And finally, the ventral posterolateral nucleus, VPL. And this is from Niedermeyer. The reticular nucleus is the pacemaker for sleep spindle oscillations. Spindles are generated within the thalamus and receive inputs from the cerebral cortex and dorsal thalamus, but also from the rostral section of the brainstem and basal forebrain. The reticular thalamic cortical reticular loop constitutes a resonating circuit that reinforces the spindle oscillations. Moreover, the spindles generated within the reticular nucleus travel along the dorsal thalamic relay pathway en route to the cortex. The brainstem and basal forebrain projections exert activating effects on the circuit with the result of inhibiting spindles. The main function of sleep spindles is the blockage of incoming stimuli on the way to the cortex. This could result from the fact that the thalamocortical neurons alternate between inhibitory postsynaptic potential and low threshold spike burst. And it should be noticed that the overlapping frequency range of spindles and alpha waves, both behavioral contexts and mechanisms, are dissimilar. Nonetheless, the same basic neural circuitry seem to have involved both kinds of oscillatory activities, sleep spindles and alpha rhythms, but working accordingly to different functional models. And that's just what we talked about earlier. SMR is one of the more common waves that we train in our field. It's a stabilizing rhythm over the sensory motor cortex and can only be called SMR over that area, though the band range it includes can be trained elsewhere as beta 1. This band range is gentle for promoting blood flow to an area, which we're going to see next in the perfusion chart, and it's one of the reasons it's helpful for migraines by gently activating an area. It's a stabilizing rhythm and is helpful for paroxysms and our epileptiform activity, and occurs when there is a decrease in activity over the sensory and motor pathways that run through the thalamus. Less attention is paid to the sensory input and decreased motor activity, and associated with a calm mental state with increased reflecting before acting. That's what most of our clients will feel, and a sleep spindle, of course. Here's the perfusion chart, and when we look at it, this is what I show every single client so they understand. A look at the red and blue lines, we can see in the delta band that there is an increase of blood flow. So when we have an immediate head injury or a person sleeping, we're getting more blood flow to that area. And then the theta and alpha bands, it drops down. We have the least amount of blood flow. That area is not working as well because that area of the brain is not needing the attention. So it's kind of turned off. It's in that resting state. And then we have it coming up into the SMR bands. It's very neutral blood perfusion. And then we get into beta where it's so much more. So when we train SMR, we're really training this neutral blood perfusion. We're really gently activating an area. So really good for migraines and for stabilizing and everything else. Here we can see an SMR spindle right at CZ. And onto gamma, trying to get everything in. So gamma comes from the neocortex, the olfactory bulb, and the hippocampus. A lot of overlap like we saw with beta, and some even with delta, as we talked about. It's for focused attention and motor task execution, response to both auditory and visual stimuli, facilitation of neuronal communication and efficient cognitive processing, and spatial working and recognition memory. And the generators are tonic activation of interneurons by metatropic glutamate receptors, cholinergic modulation of pyramidal neurons, modulation of interneurons via gap junctions, activation of inhibitory intralaminar connections, calcium-dependent glutamate release from astrocytes, and an increase in potassium to enhance activation of fast inhibitory and excitatory networks. Gamma is a neural network. Synchronized gamma frequency oscillations are enhanced with arousal and attention or by electrical stimulation of mesencephalic reticular formation. And they're mediated by acetylcholine muscarinic receptors. Thalamocortical cells spontaneously oscillate within the beta-gamma frequency range, and this occurs coherently with field potentials recorded from related cortical areas. Cortical neurons generate upon depolarization intrinsic oscillations in a range of about 40 Hz. In our field, where you can see it usually if a person is doing certain types of meditation. It's also paired with theta for learning. We're just from a bound cortical network and breathe chirps, just like we saw with that beta, so they can even ride on top of each other. But persistent, remember the whole Goldilocks thing, persistent gamma is pathological and is seen as melanocortical dysrhythmias, including tinnitus and demyelination and any other type of dysrhythmia that's going on. Altered gamma wave activity is associated with mood disorders such as depression and bipolar. For example, people with high depression scores exhibit differential gamma signaling when performing emotional, spatial, or arrhythmic tasks. And an increased gamma signaling is also observed in brain regions that participate in the default mode network, which is normally suppressed during tasks requiring significant attention. Here's an example of gamma and tinnitus. So we obviously have some muscle tension from the tinnitus, so it's a little hard to read through some of that, but we can also see some very fast activity. Once again, we're looking through some of that muscle activity, but we can see that as well. And also note the frontal theta at MC, like I talked about. So we have that frontal theta and that gamma paired together. It's very stereotypical right here for that. Finally, I want to really quickly just end with this because you will see paroxysms in your practice, and I want to make sure that we have a little slide on it. So they're commonly seen in a drowsy, unstable EEG, and they occur between state changes from wake to drowsy. They can include epileptic or discharges, but they don't have to. And it's when there's a greater than 50% of the background activity and it has a sudden start and stop. So when we see this, you don't have to panic. We just note a lot of times it happens in a drowsy, unstable EEG. And so we want to note it. We might see some other things and that adds to it, but otherwise it's just going to be our indicator that the person is drowsing and has an unstable EEG. And we can see SMR as a stabilizing rhythm for paroxysms here. So we can see that the SMR is compensatory, followed by a spike in slow paroxysmal discharge in the temporals. Most of the spike activity is from the left, but the slow is from both sides in this example. Epileptiform activity can come from a long list of things, including head injuries, autoimmune histories, family history, and much more. The slow content we see can be a cause of the epileptiform activity, and sometimes this is seen in the form of paralysis. This particular person is Todd's paralysis. So that is just the start. As you can tell, there is so much more. Each single one could go on such a topic, but I wanted to, and I even did have to leave out some of the things or even deeper in the generators that start these process, because there is just not enough time to fire hose this all at you. So finally, there is more studies that are going to be coming up. And this is really good because the QEEG course that they offer really goes over a lot of the brain waves and some parts of their generators. This goes much more into depth of it, but they do a really good job on that. So I will end on that note and open it up for questions for our final bit. And it looks like I did time it out for 52 minutes. I'm very proud of myself. Yeah. So the person that asked, how often does referring a client to a neurologist happen? A good amount, actually. So I personally, you might end up liking just to work with a neurologist. We have a couple neurologists in our field. Robert Turner is one person. He is in our field. He's really great. Some people will send every single report to a neurologist just to rule it out and to not worry. I personally, just when I see something, I will send it to him. And I've set up a thing where I pay a certain amount and send him the report. Other times clients want to go to their own neurologist. They have somebody, I would say, if I had to maybe take a guess, maybe 1 out of every 10 clients that I see, there's something that I might want to have them look at a neurologist for, or go to a sleep study, or kind of look in a little bit deeper. But it also just kind of depends on who you're seeing, and what your practice is. But a lot of people will have epileptiform activity that they were not aware of. They might come in having, rage outburst, and then we see that they have epileptic activity going on over their emotional gating. Or as, I got, examples that I showed of a client that had a brain tumor, was coming in struggling with memory. So it kind of depends. If you're working with all the athletes, you probably won't see it as much, although maybe with head injuries. But yeah, hopefully that answers your questions. I know it's pretty broad. Okay, so I'm reading, how long did it take you to be able to identify the various aspects of the EEGs? Forever. I'm still always. So for myself, I have been doing this for maybe, let's see, coming up on 10 years now. And on top of, my PhD being in this, I also have extensively, maybe every week, I sit in on a 2-hour class going over ERPs with Yuri Kropotov. And so we go over event-related potentials and EEG. Every week on Mondays, I go over for an hour with Jay Gunkelman going over the EEG. I sit in on case studies. I am constantly working with a whole bunch of other people and listening to what they're doing, listening to other talks, reading all the, latest studies coming out, as well as Niedermeyer all the time. It's a really rough book to read, hence why we started a group to have all of us read together. But it's kind of a good source of all the information there. You will never be done. I even took a whole class looking at how we can understand the EEG from a neurologist perspective. There was an online class to take. Those from South Africa. It's really good. You'll never be done. I apologize for that information, but it's a never-ending learning curve. And I personally find myself that I'm always attracted to learning things that have no definitive answer and you'll never be a master of. I think there's something really nice about that. And I also will show, I have my reference slide right here, if that, anyone wants to look at any of those things as well. Any other questions in the last minute? I know it was a firehouse of information, and I apologize for that. And that was with me cutting a lot out, but you can see each one of these could really have its full own talk on it. The EEG is really amazing, and you know, the more we learn from it is pretty great. So, and like I said, this is designed, and the reason I dove into this is so I could also understand it more because it's so confusing. You hear theta is good for learning, and then in our field, we're like, theta is bad, theta is bad. So I wanted to understand it more. So that's why I started this whole talk. And I do want to create it so all the different methodologies are put into one space. So we start understanding like, oh, well, what's this mean from, this person's perspective or this one's, and inciting them correctly, or saying if we don't have any research on it, but it's a clinical observation. So we can be correct when we tell our clients things. Thanks, guys.